sentence, Ram has red cap. Ram has red cap. This is a sentence which in which all the triplet codons are present. Ram is a triplet codon, has red cap. It makes a sense. That means the protein synthesized is, it is required for the body. Okay, this makes a sense. Now, what happens if some mutation takes place? That means if we insert a letter B between has and red, if B letter gets inserted here, then what happens? If B letter is inserted here, the triplet codon will change and we'll get Ram as BRE. In place of red, we will have this word now, and EDC and EP. This is what we will get as a result of insertion or addition of a nitrogenous base. Okay, then if three letters are inserted today, another example they have written, if three letters are inserted today here in between here, and that letter which they have given in the example is big, so it will change now. Ram has big, that means the main sentence they are talking about, not in this sentence, the main sentence. So here, right here, this only, and I'll change it now, EDC and AP, comma less. So if here only big is added, then what happens? In place of B, if here big is added, big is inserted, then what happens? Then it will change to Ram has big red cap. So see, the entire sequence changes. Because of mutation. So this is another thing which is going to happen. Now, if the letters are deleted, deletion takes place. So here they have deleted R first, RED they have deleted. If in this sentence this is deleted, RED, if this is deleted, then what we are going to have? Or in this sentence or here, so we'll have Ram has cap, just Ram has cap. That means mutation has taken place again. Now, if we delete one by one, that means if R is deleted, then in that case, what we will get? We will get RAM, HAS. If R is deleted, EDC, AP. If E is deleted, RDC, AP. If D is deleted, Ram has. If D is deleted, REC, AP. So this way, deletion and insertion, they cause mutation, and the mutation results in the change of the entire sequence. And if it is point mutation, then that particular nitrogenous base will change, and that particular triplet codon will change, and the amino acid they code for will change. But if it is a cross mutation, entire sequence will change. This shows all cross mutation where the entire sequence is changing. Now, the next topic which we are going to start is the process of translation. The process of translation is a long process. Today, I'll just give you introduction of that process, and that process I'm going to describe tomorrow. So today, just an introduction of that, because we are left with less time. So, yeah, translation. Translation comes after transcription. That means central dogma is DNA, which undergoes the process of transcription. As a result, messenger RNA is formed. This messenger RNA undergoes the process of translation, and as a result of translation, protein is synthesized. So protein synthesis takes place. So finally, this is the central dogma. It was given by whom? Who gave this central dogma, beta? Anybody who remembers? Who gave this central dogma? Yes, bache? Who gave central dogma? I have already told you it once or two, three times I have told you. I think you remember it. Who gave the central dogma? Central dogma was given by Crick. Yes, F. Crick. Yes, Paras. It was Francis Crick. Francis Crick who gave this central dogma. And you know, transcription is the process which takes place, the site of transcription. Now let's talk about the site of transcription and translation. I'll write TRCR for transcription and TRSL for translation. So transcription takes place, you know, inside the nucleus, in nucleoplasm, inside the nucleus, in the nucleoplasm. Whereas translation takes place in the cytoplasm. Cytoplasm. In the cytoplasm, translation takes place. That means if it is a cell, it has a nucleus here, the transcription will take place here, messenger RNA form. Now this messenger RNA is of no use inside the nucleus. It has already been formed in the nucleus on the nucleoplasm. Now, because transcription has taken place, transcription takes place in the nucleus, and as a result of transcription, the messenger RNA has been formed. So what is this? What has been formed? It is messenger RNA which has been formed. Now, it has to undergo the next step, which is translation. But what is the site of translation? The site of translation is cytoplasm. That means this messenger RNA now will come out of the nucleus into the cytoplasm, and into the cytoplasm, this messenger RNA will undergo the process of translation. So translation takes place in the cytoplasm, whereas transcription takes place inside the nucleus. So this is what happens after transcription. The messenger RNA moves into the cytoplasm where translation takes place. So this is this process which we will discuss now. So basically, what is this messenger RNA? Messenger RNA, it contains hidden message, and that hidden message is for the synthesis of various amino acids. That means coded genetic message is brought about by messenger RNA. So this messenger RNA brings coded hidden genetic information or message for the synthesis of polypeptide. Now you must be wondering, what is this polypeptide? One amino acid, peptide bond, another amino acid, peptide bond, another amino acid, peptide bond, another amino acid, peptide bond, and another amino acid, peptide bond. So these are amino acids. These are amino acids. Amino acids. Amino acids. And these are peptide bonds. Peptide bonds. All these are peptide bonds by which the amino acids are joined. So poly means many. Peptide means these. Many peptides are there. So that means, what is this? This is a protein. So we can also say synthesis of polypeptide or protein. So it brings the coded message which is required for the synthesis of polypeptide or protein. Now the machinery for translation is, all the machinery is present in the cytoplasm. We need some enzymes, we need transfer RNA, the ribosome is required, and by using these machinery and the message which is present in coded form on the messenger RNA, the polypeptide chain is synthesized in the cytoplasm. That means the protein is synthesized in the cytoplasm. This is clear to you, beta? The introduction of translation is clear. We will be doing the process tomorrow because right now the time is about to get over. So if I start, I'll not be able to end it. Not be able to do even little of it. So this introduction of translation is okay. Any query, any question in this? Yes, beta. Any question?